A very good morning to one and all present here. This is the day 2 of GAN course and we'll be starting with the session today by Sir Dr. Hitesh Handa.
Good morning. So let's start. I hope you remember yesterday we were talking about stress strain, right? That's where we ended. And stress is force over area. Strain is change in length over original length, right? So today we will do another type of problem. Yesterday we did this problem, right? I hope you remember. Straightforward. Then we did the hollow cylinder one. That was also straightforward. Okay. So today, Young's modulus is equal to stress over strain. Okay. Yesterday we talked stress is force over area, strain is change in length over original length. Now Young's modulus is stress over strain, stress is force divided by area, strain is change in length over original length. So now we have a formula for Young's modulus. Please remember this or write it down because on the next slide we will do a numerical problem. So try to solve this problem. So we know Young's modulus is stress over strain. We have a value for Young's modulus 200 gigapascal. Stress is force divided by area. We have force 500 Newton. We can calculate area because the diameter is given, so pi r square. And then strain is change in length over original length. Original length is given. We just need to calculate change in length. Right? So everything is given in the equation except change in length. Anybody has an answer? I don't remember, but I think you're close. I think it is something like that. I'm not sure. It's 0.07.
Yes, it's 0.79.
Yes.
0.07.
0.07, right? Yes. Because I thought 0.7 millimeter might be 2. Did you say 0.07 or you said 0.7?
I guess I have a mistake of 10.
Okay. Because I thought 0.7 sounds little high, right? But I think 0.07 is correct. Anybody else got it? Let's wait 1 or 2 more minutes and then I'll show you so 0.07 millimeter, but it's negative because we are compressing it, right? So there is a decrease in length. So it's minus 0.07 millimeter. It's very straightforward. You just have to plug in the numbers. But if you don't know how to do it, we can talk. Just talk with me later after the session. Okay. Yesterday we also talked a little bit about fatigue, right? That how, when I was giving you example of that sole of the shoe, that how many cycles it can endure before it fails, right? So if you are putting too much stress, it might go faster, but below certain stress level, it will keep going on and it won't fail. Now, another thing we have to remember to keep in mind for metal implants is corrosion, right? If a material is corroding, of course that's a problem, right? If you have a metal implant, it's corroding, it's slowly dissolving, it's going to become weaker and weaker, and the ions that are being released, they can be toxic to the tissue and it can be harmful for the body, right? So that's another thing. But also, I think you know this, but whenever we are testing all these biomaterials, implants, we want to test them in the conditions as similar to the body conditions as possible, right? So we want pH of 7.4, we want the 37 degrees Celsius when we are testing these materials. Okay. Also keep in mind, little bit off topic, if you are testing a urinary catheter, we want to test it with an artificial urine or something, right? If you want to test your material for blood application, you want blood or proteins, plasma proteins when you are testing the material. You want to test that material as close to the real environment where it's going to be used as possible, right? Here is an example, the corrosion example, that how there is pitting and corrosion happening on these implants. Titanium and its alloys have more favorable corrosion resistance for long-term applications such as joint and dental implants. Sometimes oxide films are formed on the surface, and these oxides on ceramics can actually protect the material. So titanium oxide helps protect the material, and it also passivates the implant. So overall, sometimes those oxide layers are protective. For metallic biomaterials such as stainless steel, cobalt, chromium, titanium and its alloys, there are a lot of advantages. For example, strength and manufacturing ease, availability for stainless steel, but there can be certain disadvantages such as corrosion in case of stainless steel. For cobalt chromium alloys, high modulus of elasticity is a disadvantage, because we talked about stress shielding last time. If your material is too strong compared to the bone, that can cause stress shielding, right? This also just shows an example, and again, for your exam, I don't expect you to remember these numbers, but it's just showing that how, for example, titanium, stainless steel, these are some of the type of materials used for bone fixation and devices. For soft tissue, titanium, stainless steel, nickel and titanium alloys. Some examples, you can go through this table to get an idea of how different alloys can be used for different applications. We were talking about ceramics also. So ceramics can be, so metals is one type of biomaterial, ceramics is another type of biomaterial, and of course polymers is the third type. And we also discussed how polymers are most widely used. We will cover polymers later today. So ceramics can be metallic and non-metallic composition. They have ionic and covalent bonds. We also little bit talked about ceramics yesterday, that the plate, that if you drop it, it's very brittle material. Alumina, zirconia, hydroxyapatite are some examples of ceramic materials. This is an example, and we will talk about it a little bit more today, and we will watch a video of a hip implant later. But this is a hip implant, and it has three components. There is this cup, and then there is this ball, and then there is this stem. Okay, and these three things, they can be made up of metals or ceramics or polymers, and each one have some advantage, some disadvantage. Of course, for these type of applications, we cannot make the stem of plastic, right? But this cup can be plastic, or it can be, the ball is ceramic over plastic or metal over plastic, but the stem is mostly you want it to be stronger, right? So properties of ceramics, they are strong ionic covalent bonds make ceramics very hard and brittle. So planes cannot slip past each other. That's why it's very brittle. And also, if there is any crack in a ceramic material, it'll fail very soon. And if you are confused, just think about a plate in your house, that if it gets a crack, it's going to fail very fast. Ceramics and glasses typically fail with very little deformation. You can imagine, for a polymer, you apply stress and the length can change. But for a ceramic type of material, you cannot apply stress and now ceramic is getting elongated or something. You apply too much stress, it's going to just fail and break. They are usually electrical and thermal insulators. Ceramics are sensitive to the presence of cracks or defects, like we just said. Can be porous. So for bone type applications, this porous materials can be very beneficial because the cells can go in these pores and start growing and multiplying, and now that bone implant can fix and starts becoming a part of the bone, and it does not move very easily. That's very desirable. But porous is not usually good if you have a catheter type application. Not used as extensively as metals or polymers, and one big reason is the brittleness of the material. It can be a problem. Now, ceramics can be bio-inert, they can be bioactive or biodegradable. And as the name suggests, bio-inert are materials that retain its structure in the body after and does not induce immunologic host reaction. They are inert, more inert, right? Bioactive, they are actively working with the tissues in the body. Tissue is binding to the material, so they are more bioactive in the body. Biodegradable, they are slowly degrading. So lot of times for biodegradable materials, the goal is slowly the material degrades and the tissue is growing. But you want that speed of degradation and tissue growth to be very similar or matching. You don't want the degradation to happen so fast that tissue has not grown. Now it becomes a problem. So for bio-inert material, alumina, zirconia are good examples. Bioglass, hydroxyapatite are bioactive, and hydroxyapatite can also be made biodegradable when you are processing it. So it can go as a bioactive or biodegradable. So if the material is toxic, the surrounding tissue will die, right? If the material is non-toxic and biologically inactive, almost inert, fibrous tissue forms around it. So you can imagine if an inert material is in the body is seeing this foreign object, it wants to form a fibrous capsule around it, trying to separate it from the body, right? If the material is non-toxic and bioactive, it starts bonding with the tissue. If the material is non-toxic and dissolves, it's biodegradable, the surrounding tissue starts replacing it and it starts degrading slowly. And implant material failures originate generally at the biomaterial tissue interface, right? So the interface of the biomaterial is very important because it can be toxic to the cells, and that's where the bonding of tissue happens. Now, bio-inert crystalline ceramics, for example, alumina, somebody yesterday said that, right? For dental hip implants, alumina is useful. Zirconia can be used. Carbons are good, that can be used. Porous ceramics, we talked about it, alumina, hydroxyapatite. They are good for bone implants because cells are growing in it. But remember, since they are porous, they are weak also, right? The strength is the limitation is the strength of the material because it's a porous material. So another interesting ceramic class is glasses. And this is an interesting story. So Dr. Larry Hench from University of Florida, I think in 60s, 70s maybe, I don't remember the exact year, he was working with bioactive glass, this glass material, but for nuclear radiation type application. And he was traveling to a conference, and on that bus ride for the conference, he met somebody from army, and that army person asked him, what do you do? And he said, oh, I'm working with this glass material for nuclear absorption of nuclear radiation or something, that type of work. So he said, why don't you do something about an implant, because so many soldiers who are getting injured in the battlefield, they are losing limbs because nothing is compatible and we cannot do anything about that. So when Professor Larry Hench came back from the conference, he wrote a proposal to US Army, I think, where he wanted to test this glass material in more bone implant type application. So when they got the grant funded, they implanted this glass material, I think in a rat, and his friend, his collaborator, implanted it, and after a month or so or 2 months, he contacted him and said, you know, what material did you give me? Because when I implanted it in the rat, now I cannot remove it. It bonded with the bone so strongly that he just couldn't remove it after 4 to 8 weeks, whatever time frame he tested it for. So that's how bioactive glass came into existence for bone, because it interacts with the bone so strongly that they started using the bioactive glass for that. So now bioactive glass, it's a complete synthetic material that bonds to bone. In late 1960s, when Professor Hench started working on it, commercially available, composed of silica, sodium, calcium, phosphorus. They can be bond induced osteogenesis, regenerate bone. Okay, so there can be different formulations of bioactive glass. Some bind to soft tissues and bone, some only to bone, and some do not form bond at all. So you can change the ratio of, for example, calcium, phosphorus, silica. Their ratio can be changed, and they can either bond to soft tissue or to bone or do not bond. So you can change the properties of bioactive glass by changing these ratios. But just like ceramics, low fracture resistance, right? Another example is hydroxyapatite. Up to 70% by weight of human bone is modified form of hydroxyapatite. So it can be bioactive or biodegradable depending on the manufacturing process, and used as filler for bone defects. It's mostly used for load-free implants, and sometimes the coatings of implants can be made up of hydroxyapatite because it can help in fixation of the bone. But lot of times, you know, if you want to coat one type of material on a different type of material, the coating is not very strong, and if this delamination of the coating happens, that can be very bad for the implant. Okay, biodegradable, bioresorbable materials. So do you guys know can you think of a bioabsorbable material? It can be anything. It can be polymers. Do you have any example that you have heard of a biodegradable material? Suture stitches. What are mostly made? What are mostly sutures are made up of? So a very common example of a biodegradable polymer is PLGA. You must have heard, polylactic glycolic acid, right? But we will talk about it later. So, but the main thing is, for biodegradable materials, initial strength should be there when you are implanting it, and the rate of degradation of the material needs to match with the growth of the tissue that you are replacing, right? And of course, the degradation products should not be toxic, right? Whatever it's degrading into, it should not be toxic for the cells and tissues. Okay. So this is just showing an example that how, if a material is bioactive or inert, over a period of time, a resorbable material starts breaking, a bioactive material starts bonding to the tissue, and if it's a very inert material, it's going to stay there for a long period of time. This is just another example of a study. Here is a paper if you are interested in it, that how there was an infection in a bone, and they removed the infected part and they put some biodegradable antimicrobial something, we can read it from here, filled with calcium sulfate pellets with gentamicin. So they put antibiotics in those fillers, and then over 4-month period those fillers degraded while releasing the antibiotic, right? So little bit off topic, we will talk about it later, but sometimes the problem is even if you make an antimicrobial material that's not biodegradable, it still won't release all that antimicrobial drug that's there. You know, it's there, it has the drug, but it might only release 10% or 20% or 50%. It's not releasing everything. So at that type of applications, biodegradable materials can be very good because they'll slowly release, but ultimately when the material completely degrades, it has to release everything when it's breaking and releasing it. And this is a good application. See, we cannot make a biodegradable catheter, right? But in this type of application, yes, biodegradable is okay. Slowly bone is growing, infection is going away, material is degrading while releasing that antimicrobial drug, right? So for ceramics, there are a lot of different applications. Dr. Hensch did a lot of work with bioglass type work. Ceramics and glasses are used in hip implants, mid-ear implants, heart valves, lenses. Alumina is bioinert ceramic, has minimal tissue response, very stable in vivo, typically used for prosthesis, joint replacements, low friction with little wear, right? It's a ceramic. Ceramics and glasses do not undergo corrosion like metals, but they are susceptible to other types of degradation in physiological conditions. Rate of degradation is important. Mass loss per area depends on specific surface area. Also remember, if a material is porous, since the surface area is so high, degradation can happen faster also, right? Time dependent decrease in strength happens. I think we already talked a little bit about it. So you have to be careful about biodegradable materials, how fast they are degrading. So advantages, disadvantages of ceramics: they are dense and hard materials, scratch resistant. Again, you can think about that plate. Usually they don't scratch very easily, right? They do not corrode very easily, can be polished, low friction, coefficient of friction. But disadvantages are they are brittle, they fail under very low stress, they have poor tensile strength. You can think you cannot apply stress and it get elongated. They can fracture very easily and can be difficult to machine. And think in terms of a bone implant or a hip implant. If you have a ceramic somewhere, it can crack very easily, right? Yes.
So ceramics are usually heavy as well.
Yeah.
So I mean, how does it go with the part they're replacing? Because in body,
So they are not making the whole implant of ceramic also, right? They are just like that. We will show. I'll show you that hip implant a little bit after a few slides. That how only one part of that implant can be metal and another part can be ceramic or polymer, Yeah, actually in 1 or 2 slides it's coming. So ceramic versus metals, high strength, higher hardness, superior wear, no corrosion, but very brittle, leading to fracture and hard to machine. Hip implant. So if you see here, there is like this cup, there is this ball, and there is the stem. We will watch a YouTube video also that will make it even more clear how this goes. So these materials, they can be, for example, polyethylene or metal. That cup, this head or the ball can be alloy or ceramic, and this stem is mostly titanium or alloy. They cannot be polymer, of course, right? You want a stronger material there. Lot of time these hip implants are metal on polyethylene, metal on metal, ceramic on ceramic, but they all have their own little bit pros and cons, right? Metal on polyethylene is least expensive, but it has a high wear over time. Metal on metal, it's oldest, FDA approved, and wear is very little. Ceramic on ceramic, it can be squeaky, it can be brittle, so if somebody falls, that fracture can happen. So common hip implants, and this is the FDA website, you can go on it, it had good information. Metal on polyethylene, so ball is made up of metal and the socket is made up of plastic. Ceramic on polyethylene, ball is made up of ceramic and the socket is made up of plastic. Ceramic on ceramic, ball and cup are both ceramic. Ceramic on metal, ball is made up of ceramic and socket has metal lining. Lot of time it depends on the age of the patient. Lot of times it depends on the surgeon to surgeon, what they prefer, what they feel comfortable, or what their experience is, that what type of implant is good for what type of patient. And like you can think of the risks of these surgeries, there are so many different risks, right? During the surgery there can be lot of risks. Wound can get infected, excessive bleeding, somebody can get a heart problem if they have high blood pressure, but then it can lead to blood clots, hip dislocation, bone fracture, joint infection, which is another big huge problem, because lot of time what happens, if somebody get a hip implant or any bone implant or surgery, it can take few days for the infection to develop, and then they do give antibiotics. So my father had a surgery a few months ago, and even few days before the surgery they started giving him antibiotics, and then few days after the surgery he was still getting antibiotics, right? But infection can still happen, and if somebody has an implant, it gets infected, lot of time they have to do the surgery again, remove the implant, clean that area, and then put a new implant. And it can be very messy, because I'll show you a surgery video soon. If they have to clean, so let's say they made they have 1 centimeter bone removed to put an implant. If the area gets infected while cleaning it, they might have to scrape more of that area, and now it becomes 1.3 centimeter. Now they have to put a bigger implant. So you can imagine the pain and resurgery and money wise, funds, it's a complicated situation for everyone if these gets infected. It can cause local nerve damage, numbness or weakness, device loosening. So you will see in the surgery, then they put this stem in the bone. If it gets loose, that can be a huge problem, right? You want the bone cells to grow and make it strong over time, because if it gets loose, that can be a huge issue for the patient. If the surgery is not done very properly and the implant is not implanted well, the length of the legs can be slightly different. Bone loss can happen, or stress shielding can happen. I think yesterday we saw a picture I showed you, that how when somebody had an implant and over time there was a little bone loss because of stress shielding. And then of course, if this implant is releasing some ions and they are toxic, they can be bad for the local environment, or systemically it can go and can be toxic for the body. But after few months of surgery, still it can lead to pain in the hip or leg, swelling near the hip joint, limp or change in the walking ability, and noise or popping or squeaking can happen, which is little bit indicative of that something is not right. So that can be concerning. Also, because this hip implant is so important, lot of time I have seen in old age, if people get this hip problem and the implant, they need to get implant, it can be very actually risky for life. I know several people who passed away when they had a hip implant in the old age, you know. So it's still, after so many years, so much research, still little bit of an issue, you know. Alumina in load bearing application, basically it's just showing the time dependent coefficient of friction and wear of alumina versus metal polyethylene hip joint. So over time, if you see here, the wear is very low compared to metal polyethylene. So the long term coefficient of friction of alumina joint decreases with time and approaches the value of normal joint. So that can be very favorable, that it's not, the coefficient of friction is very low. This leads to wear of articulate surface being nearly 10 times lower than metal polyethylene surfaces. So the wear is so low, which is great, right? Because you can imagine if somebody has a hip implant and there is too much wear happening, that can be an issue for the patient. So ceramic on ceramic composed of hard smooth surfaces show less wear compared to metal on metal or metal on polymer. Pros: it generates less particulate matter resulting from wear of the joint compared to the other metal and polymer. Good option for younger patients because the wear is not too much. But cons: currently only have laboratory tests and it has demonstrated catastrophic failure in very small number of patients because it's ceramic on ceramic. So it can be, so this is something that's will be deciding how to decide which type of implant for which type of patient. Okay, so next slide is the surgery. Now I think we can start polymers, metals, ceramics, now polymers. Okay, so polymers, have you guys taken some classes on polymers before? Any class on polymers? Do you guys know anything about polymers? Oh, okay. But you know what is a polymer, right? Okay, like all these plastics that we can see around, it's polymers, right? Most widely used material for biomedical applications. They are low cost, lightweight, ease of processing, flexibility, biocompatible, and I'll show you later that how just by changing little bit reaction, you can just keep changing the properties of a polymer. That's why they are so easy and widely used in medical devices. Global market for medical polymers, 5 million tons expected to exceed, this is little old information, 7 million tons by 2020. So just very widely used product. They can be non-disposable and they can be disposable products. So for example, disposable can be syringes, labware, tubing, blood bags. You just use it and throw it, right? So another big field not related to what we are talking about today is the pollution caused by all these polymers, right? That's another negative of this. Non-disposable are more that are implants for long time applications. 80% of the polymers used in medical devices are PVC, polypropylene, polystyrene. They can be used in a lot of different applications. Drug delivery, tissue engineering, vascular grafts, catheters, contact lenses, coatings, sutures, diagnostic aids. Another thing with polymers is, see, having a metal that's drug releasing sounds very complicated, but a polymer that's drug releasing is very common, right? You can make polymer coatings or films or catheters or whatever, and slowly it's releasing drugs, right? Another thing about polymers can be, if you think, they can be very hydrophilic or they can be very hydrophobic. They can have very high water absorption or they can have very low water absorption. And why is it important? So you can imagine if you put a drug inside a polymer coating, if moisture or water is not going inside, maybe the release of your drug will be very slow or maybe negligible. But if you have high amount of moisture going inside that coating or polymer, it can now interact with that drug and it can help in releasing that drug. So you can do so much of this, and we will talk it over the next few days about why this hydrophobic, hydrophilic, high water uptake, low water uptake makes a big difference in all these medical applications. So polymers have, so polymer is made up of poly and mer, right? So poly means many and mer means repeating units, right? So, and the number of these repeating units make a big difference. So polymers can have a backbone that is, let's say, made up of carbon backbone. If you have very few carbons, you can have a simple gas or a liquid. But if you have more and more carbon bound together, you can start making a viscous liquid or a simple solid type material. So you can just start as you understanding now that why polymers or polymer field is so fascinating. You can play with the length of the chains and the properties change. You can covalently bind something and the property of that material will change. So like I said, poly means many, mer means repeating unit. Polymers are lot of these repeating units bound together. Polymers are made up of monomers. So those repeating units are called monomers, and you can combine these monomers together to make a polymer. And the reaction that makes monomer into a polymer is called polymerization reaction. This is like a backbone of polymer. So carbon, 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 these are the backbone. And now you can have pendant groups. If you change the pendant group, you're just making a new polymer. So polyethylene, you change that hydrogen to chlorine and now you have PVC. A very different polymer, very different properties. You can use it for different type of applications. You change it with methyl group and you have polypropylene, and now it's a very different polymer. So for polymer chemists, this is easy. stuff, like they can easily play with the properties of a polymer and change the properties of the material. Now you can do even more with the polymers. So you can have a homopolymer with just same repeating unit, or you can make a block copolymer. So you have block of one type of material like AAA, BBB, AAA. So you can have different blocks combined together. You can have alternating AB, AB, AB. You can have random polymer, or you can have grafting, that it has one backbone and then you graft something else. They all are made up of A and B, but they all have very different properties, and you can use it for different applications. Okay, some might have a high water uptake, some might have a low water uptake, some might be semi-crystalline, some can be more crystalline. So you can just change the strength of the polymer, properties of the polymer, water uptake, hydrophobicity, hydrophilicity, all that is lot of polymer chemistry field. So common polymer biomaterials, polyethylene, PTFE, polypropylene, polystyrene, and you can just see all of these are just made by changing one pendant group. Polypropylene, PVC, polystyrene, PMMA, you're just changing up one pendant group and you're just making a new polymer. Okay. This is PDMS, we will talk about it later, but you can note that other polymers have carbon backbone. This one has a silicon in the backbone. Okay, so those are organic polymers, that's an inorganic polymer. I'm sure you probably work with these polymers, right? Do you guys work with the polymers? Oh no, okay. Nobody works with the polymer? Okay, okay. There are a lot of natural polymers. So the polymers can be natural or synthetic. See, if I knew that you have not done much polymer, I would have put little bit more meat on polymers, but okay. They can be natural polymers or they can be synthetic polymers. You know, PVC, polypropylene, polyethylene that I was just talking, they are all synthetic polymers. But you can have cellulose, gelatin, alginate, chitosan, they are all natural polymers. Okay, mechanical properties of these polymers, they depend on this chemical composition, monomer, backbone structure, size of the side chain. So you know, you have just a linear polymer or you have some side chains going on. That will change the mechanical properties of the polymer, right? Cross-linking, are these polymers cross-linked with each other? The molecular weight of the polymer, all this will change the properties or mechanical properties of the polymers. They are poor thermal and electrical conductors and they have lower strength than metals and ceramics, of course. Okay. Okay, let me ask you a question. Let's say, you know, I talk about this side chain. So let's say I have these polymer chains. Okay, this is one type of polymer. The other polymer I have is there are these polymer chains and then there are side chains. Which one do you think will be high density polymer and which one will be low density polymer? So one, there are these chains without side chains. The other polymer has this polymer with some side chains going on. Which one will be low density? Which one will be high density? Think. Okay. Let me ask. Okay. The polymer with the side chains, how many of you think will be high density? Okay. polymer with side chains, how many of think will be low density? Okay, why? So you can imagine if there is the simple polymer chain, you can stack lot of them together, compact, right? So it can be very high density. But if there is a chain with all these side chains going on, when you try to put them together, they are going to interfere and won't be able to put them together. But yes, intuitively you guys are right. That's the first thing that comes to mind, that yes, of course this is high density. But no, the side chains will make it difficult for it to become high density, right? Okay, polymerization reactions. Okay, so so my undergrad was in polymer science and chemical technology. So polymerization was a whole semester course. Okay, I don't remember most of it now, but the two simple types of polymerization reaction, one is called addition polymerization, one is called condensation polymerization. Okay, addition polymerization is very simple. You can think of like you have a polymer and more monomers are binding to it. You are adding monomers, and it's like a addition polymerization reaction. Condensation polymerization reaction is two monomers are reacting and a byproduct is forming. Okay, so hexamethylene diamine and adipic acid forming nylon 6,6 and water as a byproduct. It's a condensation polymerization reaction. Polyethylene formation is an addition polymerization reaction. Okay, so for all these polymers, there is a step that is called initiation of a reaction, then propagation of a reaction, and then termination of a reaction. Okay, if you are letting the reaction go too long, that's a problem. If you are not letting it go too long, that's a problem. The chain length gets impacted, the material is becoming more viscous or it's becoming more dilute, that gets impacted. So this is all what polymer engineers try to understand: how long the reaction should go, how long do you need to propagate it, when should you terminate it. That's all this polymerization and polymer chemistry. Okay, so just showing you initiate a reaction, you propagate it, and then finally you terminate it to make your final product. Condensation, you have this byproduct, addition reaction, you just have this addition. These are different ways how to represent a polymer. You know, you have carbon, 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 let's say polymer going on. You can have N group showing how many repeating units are there. So different ways to show a polymer material. Okay. Now, like we said, you have this pendant group, you have polyethylene, pendant group is substituted by methyl group, and now you made polypropylene. Okay, that methyl group you substitute, or this hydrogen group from polyethylene you substitute by chlorine, you made a PVC polymer. You can make PTFE. Now inorganic polymers, right? If they do not have this carbon backbone, now you have this inorganic polymer with silicon backbone. Okay, PDMS or silicon rubber, it's actually, we use it a lot for a lot of different applications. Has anybody worked with PDMS ever or seen a film or something? Okay, so one thing about PDMS polymer is it has very poor mechanical properties. It can tear very easily. Okay, but another good property of PDMS or silicon rubber is it has very good gas exchange properties. Okay, so why we need gas exchange property? Where can we use gas exchange property? Oxygenators, right? So that's one good property of PDMS type polymer. So this is what we can do with polymers, and this is like the very core polymer chemistry type people who do lot of polymerization reactions and understand, but they can just do all this, and you can imagine all this can make so many different properties of polymer. Some of these synthesis can be very straightforward, some of the synthesis can be very complicated, you know, but you can make different shape polymers, and of course they will have all different properties. Yes.
So why is this few strokes are thick and the others are thin?
Oh, actually, I'm seriously not sure.
But just thinking it
yeah.
indicates something like where
It might. So I think, like, for example, if you're making a block polymer, right? So maybe they are representing two different polymers.
that's what I think.
Yeah. So maybe actually, see, we have said B2A B2. So I think that thickness is showing B, and then the thin polymer is showing A. So actually, yes, I didn't look at it, but like, you know, AB2. So this is A and that is B. So it's like two different types of polymers being used. Now, another, so I have met him, so I was lucky to meet him once. He has done a lot of work on dendrimers. So dendrimers has this type of structure, and you can imagine you can put a drug in that dendrimer molecule for drug delivery type applications. So Dr. Tomalia, I think maybe 20 years ago, 15 years ago, he did a lot of work on this. I met him maybe around 2012. A great guy, just very good researcher, lot of citations, and he has pioneered this field of dendrimer technology a lot. Now, polymers you can also think of like these random chains. Okay, thermoplastic polymers are built so their molecule consists of many thousands of atoms arranged in long linear chains, but they don't have to be long straight chains. Like, think of a noodle, right? Also, you know, when we were talking about you initiate a reaction, you propagate a reaction, you terminate a reaction, I hope you can imagine that all the polymer chains are not going to be of same length, right? There will be some difference. You cannot just so accurately make all the chains of 100 monomers. There will be variation in a polymer. Polymers can have hydrogen bonding, van der Waals bond, they can be linear like we take, they can be branched, they can be cross-linked. You can also imagine if it's a cross-linked polymer, it's going to be very strong, right? It's cross-linked. They can have networks, chains can be entangled. You can just imagine all this happening, okay?
So cross-link would be of low density?
If they are cross-linked, so a good example of a cross-link, we will talk about it, they are like PDMS can be, and they are more thermosetting polymer. We'll talk about this soon, very soon. Okay, so now this is little bit more, little bit tricky, but it'll be okay if you just listen carefully. So polymers can be amorphous or they can be semi-crystalline, okay, but the polymers are never 100% crystalline, okay. And how you can think about polymers is, you can imagine spaghetti or noodles, and some are cooked and some are uncooked. Cooked noodles are amorphous and uncooked noodles are crystalline and they are mixed together in a bowl. So that's a semi-crystalline polymer. 100% amorphous polymer does not have uncooked, it's all cooked noodles together. Okay, and let's read from here, but approximately there are next few slides just talking about semi-crystalline and amorphous polymers. Amorphous nature of polymer is analogous to plateful of spaghetti, loose and randomly coiled. Crystalline is more like uncooked. Chains are tightly bundled and ordered. You can imagine all the uncooked part is more ordered and amorphous part is more random or amorphous, right? Polymers can be either amorphous or semi-crystalline. Polymers can never be completely crystalline. High density polyethylene molecules pack closely together, leading to a high degree of order. This makes it stiff and dense, and it's used for more like milk bottles or something. Low density polyethylene, numerous short branches interfere with the close packing of molecules, so they cannot form an ordered structure. Right? Low density, high density. You can little bit think about it, right? I have multiple slides saying this again and again, so you can like imagine it. Amorphous, loose, randomly coiled regions. Crystalline, tightly packed, ordered with high degree of compactness. Right? You can also think when you have uncooked noodles, you can put it in a small packet, but when it's cooked, you need a bigger bowl for that cooked noodle, right? Okay, X-ray diffraction, diffraction scanning, calorimetry, they are used to measure crystallinity of a polymer. Okay, higher melting and softening points, we will talk about this very soon. This just shows how in a XRD, if it's a crystalline material, what type of peaks you are seeing. If it's semi-crystalline, what's happening. For amorphous, what's happening. So you can do XRD type, you can use XRD type technique to measure if your material is amorphous or semi-crystalline or more crystalline. And you can also imagine as the crystallinity increases, there is more structure, there is more strength. You need higher melting point, right? Denser material. If you have more crystalline components, it's more dense material. And increased solvent and chemical resistance. You can imagine if you have uncooked noodles, it's more difficult to, you need to heat it more, you need to dissolve it, you need more solvents to dissolve it rather than a cooked noodle, and less permeable to gases. Amorphous, it's easier for things to move. Compact, uncooked, crystalline, it's difficult for things to move, right? Again, example, high density polyethylene versus low density polyethylene. And this is what we were talking earlier, that how if you have a high density, you have a chain, you can put lot of these chains next to each other, make it crystalline or highly dense. If you have side chains, it's difficult to make it so compact and difficult to make it so hard. So you have a milk container and more a plastic bag or something. Lower tensile strength, and yeah, you can imagine. Okay, now a different concept. Let's talk about this concept in the next session. Let me skip this and we will come back to it. Okay, glass transition temperature and melt temperature. It's a little bit longer topic. Let's go to this one. Which one is a alternating copolymer out of these two? So the first one is called alternating, the second is random. It's just random, right? Okay. Consider the following polymer chain. Which chain would you have high probability of forming crystalline regions? Why? There is a more structure to it, right, than the second one. So that's why. Okay, then let's not do this. Okay, I think now a lot of slides are this. Okay, I don't want to start talking about glass transition and melt temperature yet. Yes. So when we are testing these polymers, ceramics, metals, so always we have to first, before we do animal surgeries, we want to make sure that material is safe, because see, animal surgeries, you're compromising an animal, you don't want it to be wasted. And second is, these surgeries can be very expensive, right? So always, whenever we do these animal studies, we do lot of in vitro studies first, okay? And it can mean so many different things. So for example, when we work with catheters, let's say, we want the tensile properties tested. For example, if you take a polymer material, it has certain tensile properties. Then when you load it with drug for whatever application, are your tensile properties changing? So you need to test tensile properties. Now, is your drug leaching out? We need to that tested. We want the drug release to happen, but at certain rate, right? If we make the drug release too slow or too fast, that's a problem. If lot of drug is leaching out, it can be toxic. If it's leaching out too fast and you want it to go for a month at certain rate, but you are releasing it at like twice the rate or 3 times the rate, then you won't be able to go for a month. It might only last for 15 days, right? So that's one type of study. And we are going to talk about this later also, but since you asked, I'll answer some of your questions. Another study we should do is, there are so many, but I'm just giving you some examples. Tensile sterilization. All these materials, before you implant them, they should be sterilized, okay? So some materials, when they are sterilized, they lose strength, or whatever drug was loaded in your material, because of sterilization method, now that drug is gone. So you do sterilization and you test everything again, because before implanting you are going to sterilize it, then you implant it. But what if after you sterilize it, now the material is not strong enough, right? Or what if that drug that was supposed to be there, 30% of the drug is lost because of that sterilization method, right? And most always when we do these in vitro studies, we do them at physiological conditions, right? Because we want 37 degrees pH, whatever that application is going to be later, we want to mimic it as close as possible. You know, what other tests? Can you guys think of other tests? What should be done?
So I can think of, like, if you are for the in vivo experiment, I would go for inflammation markers first, like how it is reacting.
If you think your material is going to be toxic, right? Even if you don't think it's going to be toxic, we should always do toxicity study with cells, right? Because if it's going to come in contact with tissue or something, we should try to do in vitro studies to measure the toxicity. Is it releasing, if the drug, whatever amount you are releasing, is it killing healthy cells now, right? Another thing is, if we are doing bacteria, if you are saying, oh, my material is going to be antimicrobial in animals, we should do bacteria study in vitro first to show it's antimicrobial before we test it in the animal model. So whatever tests you can do, you should definitely do. Of course, your in vitro data sometimes is not going to match with your in vivo data. This is another challenge, because lot of times in vitro looks certain way, and when you test it in the animal to animal variation happens, and that can be a huge issue also. So, and yeah, we will talk about all those, some example of in vitro studies, bacteria, blood later, but we have to do all that. Animal studies, it's very tricky. So there are like 35 people in our group. Maybe more than 30 don't want to do animals, so you need certain type of people who can do animal studies. Like a lot of students say, I'll do everything, but don't ask me to do animal surgery. So yes, a lot of people feel sick when they are working with animals. When I started working initially, I used to feel sick, but it just kept getting better, right? But I also know some people, they just absolutely cannot do it. And in the surgery, you must have also seen how everybody is so properly sterilized. They have these gowns and everything is, because chances of infection are so high during these surgeries that they have to just be completely do it in a very sterile environment. All the instruments they are using, all the sutures or whatever they are using for surgery, it's all sterilized. The whole room is as sterile as possible. And even then, infections do happen. We will talk about those later, but hospital-acquired infections is a huge issue. Even when everything is being done so properly, so accurately, with so much care, it still happens. This is human surgery we were showing, but even for animal surgeries, when we do studies, it's almost this level of sterility. Because if we put a catheter or whatever implant for 1 week, 2 weeks, and if infection happens, it's going to like kill our study basically. So we try to be as sterile as possible, like them.
So the surgery, these kinds of surgery takes place at the same room or same designated place?
No, so yeah, very good question. So in my university, I don't know, do you guys have any surgeries here and on? Okay, so even in our university, there is only designated room where the surgeries can happen. Okay, and you have so many requirements, so much paperwork to get things done in a very proper way. So a surgery can be a terminal surgery, like you did something and the animal was euthanized the same day. Or it can be a longer term surgery where you implanted something and now you are going to look at the animal over the days and it's going to go longer. Those surgeries, the survival surgeries, are even more tricky, because if there is a surgery where the animal will be euthanized the same day, infection is less of a concern, right? You're going to do the study and after 2 hours, 4 hours, 8 hours, just going to euthanize the animal, right? So there the requirements are a little bit less strict. But wherever you want to do a survival study, animal is going to last for a week or 2 or a month, there needs to be even more specialized rooms where the chances of infection are very low. I have more class on this, but any animal surgery, everybody is so careful. we need to get so many approvals. You need to justify everything from what species of animal to use, a male or a female. I even had to write a justification why there will be one rabbit in a cage and not two rabbits in a cage. So you have to give that level of explanation, that how many animals, why do you want to use 10 animals? Why can't you do with 5 animals? If animal has fever, what are you going to do? Everything. If fever goes above this, what will happen? Because they want one, proper justification why you are doing animal study. You should have clarity. Can you get the work done without animal study? Can you get the answers without animal study? And if not, then fine, you convince that yes, you need animal. Why do you need a rabbit, or why do you need a pig? Why can't you use a mouse? You need to have that justification. Then why do you need 20 mouse or 20 rabbits? Why can't you use 10? So minimize the number and minimize the pain and distress for the animal. Like how you can do the work. Yes, we understand you sometimes need to use animals, but how can you minimize the number, minimize the distress, minimize the pain? And if even then you have to do it, if the pain becomes too high and you cannot do anything, do a proper euthanization of the animal, so animal is not suffering because of your study for too long. So all this is very, very, very strict. I'm sure in India also, in US they are very strict, so that everything is done in a very, very ethical way. You are not just randomly compromising animals and doing studies.
And so these toxicity studies which we conduct in animals, so sir, how are we so sure the results that we're getting inside animal model is going to work the most similar way in the human body? Because animal microbiome is totally different than human.
Yes, very good question. This issue is there. Lot of time we try to use the animal model that's as close to the human physiology as possible. But yes, you are still right. It can still be issues. See, in vitro to in vivo, there can be difference. Then in vivo to actually take it to the human, it can again be issues, but we need some way to go as closer and as confident so that we can just now start doing that, right?
So can cell lines be used in anything?
In
Cell lines
can be used in.
used for testing
Oh yes, of course. So when we do in vitro studies, we use certain cell lines to do in vitro studies. Whatever we feel is most close to the actual application, we test it in those cell lines in vitro to get as comfortable and as confident as possible before we go to the animal model. Because we want to be as comfortable, but we understand sometimes you cannot get all the answers and you need to move to the animal study, but just do your best so that you have the highest chances of success.
So do we go into the molecular mechanism as well with these polymers and everything, or do we just do the physiological testing?
Oh no, depends on the study to study, right? You can be doing histology to understand how when you implant is happening, what's happening to the tissue around it, or you are just looking at the device itself. So see, if you're looking at the infection, you might be looking at the device, did it get infected or not, but you can also be looking at the tissue around the device to see how badly tissue got infected or not. What if your device was able to prevent infection but tissue is still getting infected? So it's case to case, study to study, and see what I'm talking, some of these are not even my area. Bone and anything is not my area. Ceramics and metals are not my area. Even in polymers, I'm like this small part of this vast field of polymers in that application. So there is just like so many good people doing so much research in so many different medical devices. Some are looking at the device, some are looking at the tissue, some are understand molecular mechanism. It's just so vast. Yes. Yeah, yes.
But do researchers use human stem cells to test these implants?
I'm not sure how much they do that for the implants, but I think it depends on what implant is doing, what type of application people are looking at. Yes. You had a
So porous material cannot be used for catheters. You said in the very beginning. So what is the reason for that?
great. So let me go back and little bit change my statement. So catheter is a tubing you have going in your bloodstream, let's say, to either draw the blood or to infuse medicine, right? Now, if the material is very porous, because of the rough surface, protein and blood might start clotting on it, and that can be an issue. It's just it's working as a tubing to draw things and infuse things in it, right? That's the purpose of my tubing. But when you have a vascular graft, now that can be a little bit porous material, because when you put a vascular graft, and we will talk about vascular grafts also later, there you want the cells to grow, and these pores can be used for cells to grow, because there you are not using it just as a tubing, you want it to become more part of the body. But when we are using catheters, that porous material might increase complications for us, and we don't want those pores. It's just making the surface rougher for more proteins, more bacteria, more blood to interact and clot on the tubing and make the tubing maybe narrower. Yes, 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 yes.
So my question is not from today's presentation, but yesterday when we were discussing, there was this question on personalized implants. So there's a suggestion. I mean, I might
Yeah,
be wrong,
yeah.
but as going for like personalized implants, can we use like 3D printing? So not the industry making the personalized implants, but the hospital get a 3D printer and it can make a personalized implant as per the requirement of the patient.
So first of all, I'm not an expert in the field, so I'm just going to tell you what I think. I might be wrong or right or wrong. I have seen people proposing these ideas in past about personalized implants, where depending on the age of the patient or small height or large height or small, you are 3D printing and doing it. I'm just not sure how practical it is. It might even be happening actually, and I don't know, but I feel just getting it done in a hospital setting and putting an implant, it seems like it needs to be a little bit more streamlined. You need experts making it, and then person to person. But in near future, who knows, a company is selling this printer, training the people that this is how you will use it and this is how will you.
Yeah, I mean, we just give the details of the the measurements of the implant or something, and then
Yeah.
we can just print it out and we can use it for.
Yes.
Who will take responsibility in case something bad happens?
See, those are the, yeah.
But the bad things have like error of, you have that percentage of error in every.
See, there are a lot of regulations. Some things sound very common sense, but there are so many
Implications.
rules and implications. So I used to think, I was naive, that if a catheter is $10 and my catheter is $20, but it's significantly better, people will use it because it's so much better. But no, getting it approved by insurance companies, hospitals to buy it, it's not so easy. See, a hospital stay and everything can cost thousands and thousands of dollars, right? A catheter that's $10 and you are making a much better catheter for $20. It sounds so trivial that, oh, so what? It's fine. You're talking about $10,000. What is $10 catheter price difference is going to do? But no, there are just so many regulations and all that. And like she said, she has a good point. If something goes wrong with the implant, now who's responsible? Hospitals doing it, and this is just my guess, okay? I'm not an expert. I'm just thinking while talking with you. When hospitals are buying devices from a company and putting them in patients, at least they are not responsible for device, right? They are only responsible for surgeon implanting it, right? But now if they are making the devices and implanting it, and if it fails, now who's liable for that? So I don't even know if hospitals would want to do it. It's a great idea. Maybe, and again, I don't know, maybe patient comes and those measurements are still sent to the company and now they are still providing the device, but that will always increase the cost of the device significantly. See, right now they are making thousands and thousands and lot of catheters in bulk and now selling it like, okay, $10 a catheter. But if you want this specialized device, the cost is going to go up a lot probably, you know.
The hospitals can collaborate with the manufacturers and the R&D department of based on government, so they can work and together to just build something which can be cheap and can be useful. For, like you are saying, we have to build a catheter that is in the range of $10, and then to it is more useful than the previous, like the usually used one. So we can just do R&D on that thing, working alongside the government and the hospitals, because the surgeons also want something better so that they can help the patient in a better way, I guess.
Yeah. So you're saying a really interesting thing that's actually kind of helping in US. Lot of good universities, good hospitals, they want their MDs to do research. So proper hospitals that are under universities and even otherwise, those MDs and surgeons, they are seeing the patients, treating the patients, doing surgeries, but they have their own labs also going on. You know, when I was working in University of Michigan Medical School, lot of the surgeons, doctors I was working with, yes, their main responsibility was attending the patients, but they had labs, because if they are like, so for example, my mentor was in ECMO. Okay, he's using ECMO on patients, but while using it on patients, he's realizing a lot of limitations of ECMO or challenges with ECMO. So in parallel, he has a lab going on where he's trying to get funding from NIH and address the issues so that in future they can be, the modified material can be applied to his surgeries and patient treatment. Yes. And actually that's a good way to do it, because who knows better about those than the surgeons who are actually treating the patients every day, right? And that brings us to the point, yesterday we were talking about collaborations. It's happening in US a lot, and I think it must be happening in India also. Engineers like you guys working very closely with hospitals. See, what helped me was working in the medical school. You know, lot of time biomedical engineers are just working on in the lab, learning things, right? But if you guys work very closely with doctors and clinicians and surgeons, you get to know about the problems. And lot of time problems might be easy to solve. See, their training is in patient treatment. They are not engineers. You guys know how to solve a lot of things. You just don't know what the problems are. So by working closely with the clinicians, we can find solutions to the problems in easily. Right?
But the patient load in India is so high that doctors hardly get time to do research.
I know.
In a hospital setting,
Oh
so
yeah.
we have this experience.
It is. I think, and I don't know how this can be addressed in future, but I think US is doing this very well, and maybe in India also. I hope they can try. But you are right. If you are busy morning to night just seeing patients, you have no time to do research, right? So, okay, guys, let's start our afternoon session. So I see at least 4 or 5 new faces now. We had started talking about polymers can have amorphous regions and they can have crystalline regions. Okay, so a polymer can be completely amorphous or semi-crystalline, but not completely crystalline, right? And we were talking how amorphous parts are like cooked noodles and crystalline part is uncooked noodles. Okay. Okay, so now we'll little bit talk about TG or glass transition temperature and TM or melt temperature. It can be a little bit confusing, but it's not difficult. Also, polymers can be thermoplastic or they can be thermosetting. Okay, we talked about this earlier. Okay, glass transition temperature is always lower than melt temperature, okay, or melting temperature of a polymer. Okay, another thing to remember is glass transition temperature is associated with the amorphous part of the polymer and melt temperature is associated with the crystalline part of the polymer. Okay, so most polymers will decompose before they boil. Cross-linked polymers decompose before they melt. So cross-linked polymers, and you can think about a polymer that's cross-linked, you cannot dissolve it and make it a liquid. Okay, they are set or also called thermoset polymers. Once they are set in a shape, or let's say you made a film or a catheter with that thermoset polymer, it's now set. You cannot melt it with in a solvent or something. Whereas thermoplastic polymers, you can dissolve it in a solvent and you can reform it in a new shape. Glass transition temperature is also TG. Melt temperature we are representing as TM, are different for different polymers, but TG is always lower than TM for a particular polymer. Okay, for example, for polyethylene, PTFE, PVC, there are just some examples. Of course, you don't need to remember it, but TG is always lower than TM for the polymers. Okay, now there are next few slides I'm going to say same thing in different ways just so that you understand. You can think, and it's coming on the slides, I'm just trying to explain it. TG is below which, so below means you are cooling it. Okay, so below TG your material is becoming less easily flexible to move. The chains are becoming more rigid. So let's say you have a polymer at certain temperature, you start cooling it,